Okay, welcome to lecture 4 of the Oculomotor module, part of PSY6315, and I'm going to be talking about saccades, the wider brain beyond the superior colliculus. We've spent a lot of time talking about the superior colliculus in the past 3 lectures, or at least certainly in the past 2 lectures, and then I'm going to talk a little bit about attention and decision making. And the idea of this is really to say, well, what grabs your attention, and what are you going to look at next, and how does your brain basically decide on this? So we'll talk a little bit about where the brain is doing that. So this lecture is really aimed at directing your reading. So we're going to put the superior colliculus in context. I'll talk a little bit more about it, but also about other brain regions that are involved in eye movements. I'm going to look at decision making in the oculomotor system. I'm going to just sort of mention attention, which is clearly related to decision making, and it's related to what you're looking at. Often if you're looking at something, then you are attending to it, but you don't have to be looking at something to attend to it. You can attend to objects in your peripheral vision as well. I'll mention lots of papers. I've kind of put pictures of them on these slides. So feel free to follow those up. They're good starting points for your reading. And then I'll mention something about how to write a great essay for the exam question. So let's put the colliculus into context. These are the main subcortical regions involved in saccadic eye movement. So obviously the colliculus is there. It's involved in target endpoint specification, as we've discussed. It is involved in the dynamics of the saccade, it would appear, from the Goossens and Van Opstal paper that we looked at in detail. You can also look at their follow-up paper in which they discuss whether the movement that the colliculus is encoding is an optimal movement. That's quite an interesting paper. It's from 2012, I think, so you can look that up. Same authors. And it's involved in head and trunk movements. Superior colliculus is involved in head and trunk movements. But it's not just the colliculus. The colliculus is connected to the thalamus. The thalamus in the middle of the brain. We're going to have a look at where these regions are in a second. The thalamus is involved in relaying sensory input from the retina through to the visual cortex, but it also relays signals from the colliculus and around into the cortex and back. So thalamus is important. The basal ganglia is important. It projects into the superior colliculus and is involved potentially in target selection and is involved in saccade initiation. The cerebellum is a very important part of the brain, and it's involved in motor adaptation. Keeping your eye movements accurate. And then, of course, the lower brainstem is involved because at some point we've got to get the motor neurons, for example, those in the abducens nucleus, to fire and drive the rectal muscles to move the eye. So here's a picture, a Nissl stain. Nissl stains highlight genetic material. They highlight DNA and RNA. It's a chemical. And because in the cell bodies of neurons you get much more DNA and RNA than in the white matter, you get this beautiful stain which shows where the majority of the cell bodies are. You get a really good look of the cerebellum here with its very characteristic structure here. Up here is the isocortex or the neocortex. This is a macaca mulatta, a monkey brain. You can see these brain slices at primatebrain.org. And this is just one of about 50 slices in a single brain. Because this is a sagittal slice, so we've got the back of the brain. Here's the brain stem that then extends down to the spinal cord. This is towards the front of the head. We can see the brainstem in the middle here. and we can see this bit's called the midbrain. So this is quite a nice slice because it highlights the superior colliculus right here, and you can see the inferior colliculus below. I don't talk about the inferior colliculus, but they appear close to next to each other. Sort of in the middle, just above the colliculi, is the thalamic region, which is quite a big and important region. It connects up to the cortex and then back down to other parts of the brain down here. So it's often considered to be a sort of relaying station, a sort of wiring station. The basal ganglia is this region here. So basal ganglia nuclei include the caudate, which is this one here, the globus pallidus right here, and then we've got the substantia nigra down here, yeah, so this region around here is basal ganglia. It's this substantia nigra reticulata, the SNR, which projects into the deeper parts of the colliculus. Okay, so that's kind of cortical regions here, subcortical regions, I'm sorry. And then the cortical regions that are active for saccades are many and varied. The most, well, I say the most important, the one that we tend to talk about here in Sheffield is the frontal eye fields, which encode saccade targets in much the same way that the superior colliculus does. It's kind of like the cortical version of the colliculus. It has retinotopic coordinates. The coordinates are eye-centered, just as they are in the colliculus. Near to the frontal eye fields is the supplementary eye field. Works in a slightly different way, has a slightly different set of coordinate systems, or a slightly different coordinate system, and people have suggested that it learns the value of saccades. You can lesion the supplementary eye fields and you won't see huge deficits in eye movements. And then we've got other regions, the lateral intraparietal cortex, another bit of cortex here which people have studied for eye movements. Again, another kind of coordinate system. They're non-retinotopic, and they're not eye position dependent. So have a read about that. The visual cortex at the back of the head is involved in processing images that are coming in, and doing the kind of complex things that mammals can do with images, such as object recognition, edge detection, that kind of thing. And then the sort of dorsolateral prefrontal cortex, so sort of dorso up at the top and to the side of your prefrontal cortex, is people relate that to volitional saccades and much more complex tasks that actually involve what you might think of as thinking. So there's the cortical regions. Just to kind of give you a sense of where they are, here's it's the same monkey brain, but we've moved laterally out from the center line, so we can't see the brainstem anymore. We can still see the cerebellum here. Area 17 is the primary visual cortex. That's at the back of the brain here. It's the back of your brain as well. And then towards the top and the front is area 6, supplementary eye fields. Area 8 is somewhere around here at the front. This is a sort of sulcus that's got the interparietal cortex in. There you can see in this one the lateral genicular nucleus, part of the thalamus, is visible in this slice. That's the relay from the eyes through to visual cortex. And here's yet another slice, even a bit further out, and we can see. Oh, sorry, I said wrong. This sulcus here divides the supplementary eye fields and the frontal eye fields. The lateral intraparietal cortex is back here, so quite a long way away actually. As it goes, area 17 is again marked at the back here. Okay, so that sort of gives you a sense of where these different brain regions are. So if we're thinking about decision making in the brain, let's think about what would be the simplest experiments that we could perform with either humans or animals. the simplest decide where to look experiment. And you can't really get simpler than the two-choice decision. And in this case, that would be a case where you start off looking at a fixation point, this red dot, and you are then given choices of moving left or right. Maybe luminances in the field, they might be shapes of different types of shape. Maybe you have to decide which shape to saccade to. Maybe you have to decide based on colour or based on brightness. And it's worth noting that if the choice is between a left and a right movement, you are studying decision-making shared across both sides of the brain, because there are two superior colliculi, one on each side of the brainstem, one on each hemisphere. The left colliculus encodes objects that are in your right hemifield, and vice versa. So if you have to make a choice between the two, you will have some sort of activity potentially on both colliculi, and your brain needs to resolve the conflict between those two. Alternatively, you could make a two-choice decision task where both of the potential targets are in the same hemifield, which sort of simplifies your study and means that you're then looking at decisions that have to be resolved within one colliculus, or say one side of the frontal eye fields. But either way, so the question that I'm going to talk about here is where are two-choice decisions made? Okay, so a good question to ask the literature is there a neural substrate for saccadic decisions? And if you start searching for saccadic decisions, you will find a lot of different potential neural substrates, i.e., particular brain regions that are implicated in making the decisions. Maybe it's the prefrontal cortex. If you find this Frontiers in Neural Circuits paper, determining monkey-free choice long before the choice is made, the principal role of prefrontal neurons involved in both decision and motor processes. So this is looking at, so what they do here is essentially they're looking at the activity of neurons and using the activity of those neurons to predict which way the monkey is going to move their eyes. You might find another paper that will tell you that the lateral intraparietal cortex is making perceptual decisions. You'll find that there's lots and lots of different areas. There's a couple of cortical areas that are implicated. You can have a look at those papers and have a read about them. The way to go about reading a paper, especially when it's one that you're not sure is going to be particularly useful, is to read the abstract first, and that should incorporate a kind of description of the main result. And if that doesn't quite kind of tell you, then I often skip to the discussion and have a read of the discussion, because that generally recapitulates the results without all the gory details, so that you can get a sense of what the paper's about. And then if you then want to know more about the details, then you can delve into the results, and if you want to know the context of the paper, then read the introduction. Okay. Another possibility is you might find quite a few papers about the colliculus as the substrate for decision making. So this paper here will covers that. Covers two choice decisions in particular in macaque with the superior colliculus. There's a nice review by Michel Basso and Paul May on the role of superior colliculus in decision making, and whether it's actually really participating in cognitive tasks, even though it's a subcortical brain region. This paper here is a good one by McPeak and Keller, where they inactivate the superior colliculus and look at how saccadic target selection is affected. So maybe it's the colliculus that's doing the decision making rather than the cortex. Could be the basal ganglia. We like the basal ganglia in Sheffield. We've I've participated in work that was started with started with Jonathan Chambers under Kevin Gurney's supervision, continued on by Alex Cope, and then by me later on. It's well known that the basal ganglia is connected to the colliculus and the frontal eye fields, so there's Hikosaka has written a couple of papers on the role of the basal ganglia as relating to saccades. It's gone blank. Okay, sorry. I think those were probably supposed to come in one by one, but they've all appeared at once. That's no problem. So really, the question is, you read all these papers, they all say, my favourite brain area is the one that's doing saccadic decision making, and here's some experimental evidence to support it. Is one idea right and the rest wrong? Is this a question of, finding, the one true decision maker? Or it could be the case that one region is doing the decision making, but the rest, even though you can find evidence for the decisions in other brain regions, perhaps they are just they're just reflecting the decision that's made elsewhere, and they're just indicating the outcome. Or it might be the case that that they're all participating in the decision making, that they're actually operating together. Maybe they're making decision. That they're sort of accumulating evidence for which way you should look based on different criteria. Potentially, the cortical regions are interested in particular complex features of the choices, whereas lower level regions are doing decision making based on fairly basic features like brightness. Okay, so let's think about this. Let's just go back to the colliculus. I wanted to show you a couple of papers from Michel Basso's lab, and these are about the fact that the colliculus is really quite a complicated brain structure. It's a 7-layered, people usually think of it as a 7-layered structure. So if you look at it under a microscope, you'll see different kind of, especially if you stain it, you'll see different structure to the cells, different cell densities, different cell types in 7 different layers. These papers I'm going to point you at are nice because they show you what they show some of the dynamic behavior of the neurons in those different layers using this real-time imaging technique called dye imaging. So let's have a look. So here's the technique. These are in vitro experiments. So they've taken slices of colliculus, and then they put them on under a microscope. They put a stimulating electrode in so that they can put a pulse of current into the preparation. They've got a recording electrode to record response in some other region, somewhat distal from the stimulation. But they've also put a dye on the tissue, and that dye is voltage sensitive. So I'll just move my face up there. And that means that you can watch the voltage changing across the tissue as the currents flow through the different processes, the axons, the dendrites, and across the synapses. Okay, so it gives you real-time imaging, and you can watch the dynamics. I've just recalled that I haven't actually put the videos into this presentation, which is a shame. You can look them out. There's some really nice videos that are associated with these papers. But basically, this first paper, they looked at this is a kind of orienting you to the tissue, that you've got the top layer here, the SGS, that stands for stratum griseum superficiale, so it's the superficial grey layer basically, and then this SO stands for stratum opticum. That's a sort of I think it's kind of a more see-through layer when you look at it, although it doesn't look very see-through here. And then SGI is the stratum griseum internale, so that's kind of the more deep layers. And they looked at the different kind of responses that you get when you stimulate either in the superficial layer here or in the deep layer here. So they've dropped their stimulation probe deeper down into the tissue. And the general result is that deep stimulation projects activity up to the superficial layer where it then spreads out, whereas when you stimulate in the superficial layer you just see the spreading out. You see a sort of an initial spread and then it kind of contracts back in both cases. Okay. Again, they show these nice time series, time sequences, so that you can get a sense of the dynamics. This on the left here, panel A, is the superficial stimulation. You see the initial stimulation up here, which then builds up. It kind of gets wider and wider, and until essentially it's got to spread out quite widely. And over on this side, oops, don't want to be that big. Over on this side, where the stimulation is deep, it builds up kind of in the same way, but then it starts to float up to the top, and then you see the spread that looks quite similar to the superficial stimulation there. And they use this to sort of say that there's some interesting dynamics going on within the colliculus itself. Okay. So this is a follow-up paper that was published. It was the same first author, same group, Michel Basso's group. It was published about 3 years later. And they do some fitting to suggest that in the superficial layers of the colliculus, they're seeing a normalization process. And that they think that there's actually a sort of saccadic averaging process going on in the superficial layers. So this is a sort of recap of some of the results, but the main difference in this paper is that they stimulate in 2 locations at once, and they show that from the 2 deeper layer stimulations that they make, they get a kind of averaging of the position in the superficial layer. So those are quite nice to look at, and they give you a sense that the colliculus really is, it's more than just a simple sheet of neurons that some of the models kind of reduce it down to. I mean, we always have to we always have to simplify brain structures in models, because that's the whole point of the whole point of modeling is to try and find the fundamental mechanisms within each region. But this is some nice data to show that it's actually quite a sophisticated brain region in its own right. The Basso lab, or Michel Basso, has also written papers about attention and the colliculus and decision making. This is not one of hers. This is a paper by James Muller, Mario Philistides, and it comes from William Newsom's group. And they looked at microstimulating the colliculus, so not enough to move the eyes, but enough to improve attention in a particular task. So they showed that the colliculus participates in attention, and let's have a quick look at the. So here's a figure from their paper, which sort of describes the experiment. It's an experiment with monkeys. They train the monkeys to fixate on a central cross first. They then, in this region marked out in blue, they get the monkey to look at the movement of some dots. And if the dots in that region tend to be moving to the right. Then after a delay, the monkey has to saccade to the right-hand red target, and if it gets it correct, it gets a reward. And what they found is that if they stimulated the colliculus with a low-level stimulation, which is not enough to make the eyes move to that location, but if they stimulate it with this microstimulation in the area where they're asking the monkey to make the discrimination of the movement direction, then the monkeys actually do better on the task. They actually get the answer right, and they move to the correct target more often than when they don't stimulate that region. So even though the eyes haven't moved there, the colliculus is participating in some way in attention of that area and in the monkey's processing of that area. There's a similar result for the frontal eye field that was shown. So they performed this study based on a similar result for frontal eye fields, and it may be that the stimulation that they apply in the colliculus is finding its way into the frontal eye fields and the cortical regions and assisting with the cortical decision making. Okay, so that's attention in the colliculus. I've talked about the colliculus. I want to talk about the cerebellum as one of the other subcortical regions. I'm going to talk about a combined oculomotor model in a few slides' time. The cerebellum is absent from that model, but I just wanted to talk about what it's about. Again, in Sheffield, there's been a lot of thought about the cerebellum, particularly by Paul Dean. Cerebellum is very much his specialty. He's published papers about saccadic eye movement. Sorry, not saccadic. The cerebellum's involvement in the vestibulo-ocular reflex. The cerebellum seems to contain a sort of generic mechanism for adaptation in motor tasks, and because almost everything you do is a motor task, even speaking, the cerebellum is involved in just about all of the things you do. Eye movements are certainly assisted by the cerebellum, and this adaptation aspect is very important because the eyeball physics can change either with age, injury, or short term. Your eye muscles could simply become tired, and so the innovation that's required to get them to move may change over a relatively short time period. So that's a good argument for why you have this kind of specialized adaptation device, which we call the cerebellum. People have looked at saccadic behavior and the cerebellum. They've done so by lesion studies, so by inactivating or removing the cerebellar cortex. So here is a paper that covers that. After the lesion, saccades become much shorter, and there's no longer any short-term adaptation to how long saccades to be. And they study that by making saccadic targets move while you're making the eye movement. So if I put you in a saccadic eye movement task, and every time there was a target that you had to saccade to, I moved the target 10% further than it was, after a few minutes, your saccades would actually start to adapt to that additional movement that I was applying. But if your cerebellar cortex had been lesioned, you wouldn't have that short-term adaptation. A long time after the lesion, the saccades do recover to an extent. They get less hypermetric, so they start to get to their correct target, but they remain much more variable than normal, and they never regain this short-term adaptation. Another way to study the cerebellum or any other brain region is to look at pathology. If you can find people or animals that have got problems in a specific region, and this paper covers a number of patients who had cerebellar ataxias, problems with their cerebellums, and it looked at their saccadic behavior. one particular take home from the paper is that late onset cerebellar ataxia patients had variable eye movements, much more variable than normal. But you do have to be a bit careful of this one because it was only, I think, 8 patients with one disease and 10 with the other. But that's another way of looking at what a brain region contributes to function. There are models of saccadic control by the colliculus and the cerebellum. And this paper here from Quire, Lefebvre and Optcan is, I think, one of the main ones. I couldn't find any others, actually. But the authors remind the reader that saccades can proceed even with the inactivation of the colliculus, hence the need for models beyond superior colliculus. If you've got an adaptation device which adds a signal to correct the functionality of another signal generating region, potentially, if the original signal generating region stops producing any signal at all, the adaptation device could take over entirely and produce the entire signal that's required. And that's really one possible argument for how it is that saccades can continue to work with a lesioned superior colliculus. I think in this paper they refer to the original results where it was shown that you can lesion superior colliculus and saccades still work. Look that up and have a look. It's interesting. And maybe it's the cerebellum responsible for the dynamics that we looked at in the Goossens and Van Opstal paper. Consider that. So you've got these. They refer in this paper. They refer to the connections that exist between colliculus and cerebellum. So it may be that if you remember, the Goossens and Van Opstal paper shows that there are dynamics in the colliculus, but it doesn't give any mechanism for that dynamic behaviour. And there's another. I've given you a link to a review here rather than a picture as well. Okay. I want to say something about cortical regions and saccadic decision making. So this little mini module is not very corticocentric in the sense that you won't be expected to have a very full discussion of cortical saccadic control in the exam. So sort of focus your attention more on subcortical regions. But cortex is usually given the kind of higher order cognitive responsibility, and it certainly is involved in saccadic eye movements. You can stimulate frontal eye fields, you get a movement. You can stimulate supplementary eye fields, I think, and you get a movement. And it's certainly involved in saccadic eye movements. I guess the point I'm trying to make here is it's always working in concert with the rest of the brain. It's working in concert with the colliculus, the cerebellum, the basal ganglia. They're all working together. And so see those Basso papers on colliculus and cognitive tasks and that Muller attention paper that I just talked about. Okay, so that's all I'm going to say about cortical regions right now. I'm going to say something about basal ganglia and saccades, and then I'm going to talk about a model which pulls together this idea of using basal ganglia or incorporating basal ganglia into a model of the eye movement. This is the model which completely misses out the cerebellum. So it has no adaptation. We admit that we omit cerebellum, but we suggest that what we're missing from cerebellum is the ability to make consistently accurate saccades, but we're not necessarily losing the ability to make decisions. Okay, so this is work which is based on this paper by Alex Cope, which this is the one that's only available on bioRxiv. The link to it's in one of the previous slides. And I've taken a quote from a Wurtz and Hikosaka paper from 1986. The SNR projection to the superior colliculus represents one of the most prominent inputs to the intermediate layers of the superior colliculus. And that projection that they're talking about here in that paper is this dotted line that goes from this region here, BG, down to this box here, superior colliculus. So that's the prominent connection. And this is a kind of cartoon diagram of the layout of this model, which has got a colliculus here. It's got a brainstem saccadic generator here, SG. It brings in information from the world via the retina, which comes into the superior colliculus and also passes its way into the frontal eye fields, which is the entire cortex in this model, is just one map of activity. And in the middle of it all is this basal ganglia decision-making box. So the model incorporates the idea that of course we've got topographic arrangement of tissue in the frontal eye field, in the retina and the colliculus, but this model incorporates the idea that that topographic arrangement persists in the basal ganglia and also in the thalamus. Note that these dotted lines are inhibitory. So what the basal ganglia is doing is, this is its standard sort of mode of operation, which I think will be talked about in another module in this course. But the basal ganglia inhibits access to motor resources. So essentially, what it's doing is it's gating the colliculus here, and then it's going to allow a saccade to start. Okay. I'm going to point you at this paper, which we published in 2018, which considers the system as a whole. So it takes that model that I just showed you, this one here, this brain model, including the brain stem, and it plumbs it into a biomechanical eye model and makes a complete closed loop model. Okay. And the reason I want to talk about this system as a whole is it allows us to think about, if we want to worry about where decision making is happening in the brain, this will give us a kind of perspective on how to think about the experimental results that people publish about decision making in the various different regions. So the model is a mechanistic brain model. I'm going to show you some diagrams of it in a minute, but it basically consists of selected populations of neurons, and the interconnections match the neuroanatomy. So there's a lot of care has been taken with this model to study the literature and look at where the anatomists say that there are connections between different regions. And as many parameters in the model as possible are fixed using data that we can glean from the literature, such as number of connections, proportions of connections from one place to another. If you're worried about what a mechanistic brain model is, think about what a non-mechanistic brain model is. And I've written down a quote here which gives you an example of a non-mechanistic brain model. And that is, the size of a horizontal saccadic eye movement is linearly proportional to the angle from the center line made by an isolated stimulus, which is just another way of saying, you look at a stimulus. And that model models the overall effect of the brain and body without considering any of the component brain regions and what they might be doing. It's a kind of, it makes the whole thing into a black box. And in that case, you don't really need a model for that, but that is it. That's a non-mechanistic brain model. So here, back to a mechanistic brain model. Here's a small part of the model. It's the diagram. I show the diagrams as these box and arrow diagrams. So it basically shows you what populations we're considering. 2,500 is the number of neurons or neural elements in that population. This text in grey tells you what mathematical model is being used for those neurons. and then the arrows with a description of the connectivity pattern tell you how one population connects to another or to itself. That's a recurrent connection there. Okay, much of the detail in the model is hidden in this view. Okay, you can go from that same box and arrow view and visualize the whole thing, neurons and all, to get a sense of the kind of complexity of the model or the scale of the model. So this is showing the same three populations. So it's 3 sheets of 2,500 neurons. Each one is 50 neurons on a side. In this case, we've got a one-to-one connection from the retinal population to the superficial layer of the superior colliculus. And then from the superficial layer to the deep layer of the superior colliculus, we've got this Gaussian connection, which means that, so this is only showing connection from one source neuron with these green lines to the SC deep layer. But you can see that each neuron sort of sends projections in a kind of widening or a splaying pattern. So one neuron in the source connects to multiple neurons in the destination. And then these green lines here show you that there is some recurrent connectivity within the layer as well. That's this recurrent arrow here. Okay. So this is the sort of box and arrow of the brain model. And it's the same connectivity as the diagram I showed you earlier. Again, it's known anatomy, so we've got lots and lots of connections here. The connections with the little circle are inhibitory connections. So here's the inhibitory connection from the substantia nigra, the substantia nigra reticulata. That's projecting into the deeper layers of the superior colliculus. And the arrowheads are excitatory connections. And really, when you start building a model like this, and indeed running the model, this is when you realise that it's not easy to figure out. I'll just move myself up there. It's not easy to figure out where things are happening. So if you've got competitive processes within some of these brain regions, some of these maps, then the result of that competition is also projected into other regions. And so suppose I had a competition in frontal eye field, and the competition was actually achieved in frontal eye field. Well, frontal eye field is also exciting the deeper layer of the superior colliculus in this model. So I might look at the deeper layer of superior colliculus and see that the one of the targets was winning out, that the hill for the right-hand target was perhaps being was winning out and was suppressing the hill for the left-hand target by some internal inhibitory connection. But I wouldn't necessarily know whether that competition was happening in colliculus or in frontal eye fields. So note that within this model and within the anatomy, there are loops. Information is flowing around the brain in loops, and that's really the what I'm trying to get across here. Okay. Yeah, there's one other thing to note about this model. It has 2 pathways. It's got a fast pathway, which takes you straight from the world, which is just a representation of what the eyes see, straight into the retina and straight into the colliculus. So that's the kind of simplistic processing of targets. And then it's also got a slower input path, which takes you from the world into the frontal eye fields. So it misses out the pathway through the lateral geniculate nucleus and all of the visual cortex. It just says, okay, we end up with some activity in the frontal eye fields. That's what makes this model interesting, in the sense that it's balancing cortical processing and decision making associated with the basal ganglia, and a faster process. And this model does make it express saccades, the very fast reactive saccades, and also slower saccades. Okay. Okay, so yes, let's just talk a bit more about this one. So the grey box here, that's the basal ganglia model, which is actually, it was a pre-existing model which was published by Kevin Gurney, Pete Redgrave and Tony Prescott from the department, and it postulates the basal ganglia basically as an action selection mechanism. So this is sort of, it has this characteristic shape to it, but essentially it's processing multiple channels, and it's a selection mechanism between those channels. Okay. There's in the overall model, there's almost no model for cortex. So again, I said this wasn't very a corticocentric course, and that's really because the work that I've been involved in hasn't been very corticocentric. We reduced the whole of the cortex down to two boxes, one which adds a bit of noise to the world representation, and one which then projects and participates in the loops. So the model incorporates no way of investigating competition within cortical regions. So yeah, we can't look at competition within LIP. So that's just not accessible within this particular model. The superior colliculus model here, incorporating these two boxes, so it has a population for the superficial layer and a population for the deeper layer. Remember, the Vaucoon and Basso papers seem to show kind of, although it's a multi-layered tissue, you could sort of see that there was deeper stuff and then there was superficial stuff, and so this may be a reasonable kind of subdivision of the colliculus into two populations. So the paper that the collicular model is based on is from Arai and Keller, and their model correctly predicts saccade trajectory variation due to competitive mechanisms within the colliculus. So they looked at comparing their model to experiments in which saccades were made in the presence of a distractor target. Okay, so there's some quite interesting papers associated with the Arai and Keller model to do with saccadic trajectories and what that can tell you about what's going on in the superior colliculus. There's a brainstem submodel, and this is, if you remember in the Goossens and Van Opstal paper, they were taking empirically measured activity from the colliculus and driving it through a simple brainstem model. This is a slightly different brainstem model, but it's not entirely different in the sense that it's got two channels, one for left, one for right. In the whole model, there are 6 channels for the 6 extraocular muscles. But what's interesting is that even here there's a decision mechanism. It's the crossed inhibitory connections. So these lines with a dot are inhibitory, and you see they cross over, so the right is inhibiting the left output and the left is inhibiting the right output. So I say it's a decision mechanism. It's essentially a kind of sanity check that the brainstem is making on the output from the brain, and it's just preventing both eye muscles being active at the same time, which would be no good. You don't want to try and pull the eye to the right and try and pull the eye to the left at the same time, because you'll just pull the eyeball back in its socket and potentially damage it. So clearly a sort of, and this is based on known anatomy, clearly a kind of fail-safe mechanism has evolved to prevent that from happening, and that's a decision process that's happening right down in the brainstem. So what conclusions could we take from that model? Well, we can't take very many conclusions about decision making from the James et al. Paper from 2018 because we didn't actually look at decision making in that paper. We didn't present any results. But what the experience with the model indicates is that it's really hard, and we found this from debugging problems in the model. It's really hard to know where a signal sort of originates when you've got a model where everything is connected to everything else and you've got loops through the system. And the model also suggests that these decision-making mechanisms, because there are several locations where there are decision-making mechanisms in that model, it suggests that they're all working together. They might be competing with each other, or they might be reinforcing each other. But I don't think, my view is that there's decision-making going on all over the place at the same time. And that different brain tissue is kind of contributing different kinds of decision-making processes. So, right down at the brainstem, you've got mechanisms to prevent crazy stuff like trying to pull the eye in both directions. Up in the cortex, you've got much more complicated decision-making mechanisms like, I know I mustn't look at the red dot, and I must only look at the yellow dot. So you're doing much more complicated stuff in a kind of higher order part of the brain. And the other thing that this particular paper tells us is that modelling individual brain regions requires that we make assumptions about inputs and outputs from each of those brain regions. And really the main result of that paper was that you learn something new when you take multiple models and make them into a closed loop, because you will find that, or certainly we found, that there were connections we had to add in without which the model would not do what it was expected to do, i.e. make a pro-saccade. Okay, so have a read of that paper. Saccades and decision making. I think there are multiple layers of decision making going on. I sort of explained that already. This is really a recapitulation of what I just said. Low-level circuits like brainstem have a strong fail-safe decision mechanisms. Other regions might contribute to the decision making or reflect decision making in other regions. Higher cognitive level regions may act to suppress expected reflexive behavior, probably via the basal ganglia. Okay, so there we are. That's kind of my thinking about decision making and saccades. So let's just finish up by discussing how to do well in the exam. So the exam these are essay questions, and I find it's really helpful to try and explain to you kind of what you're expected to do. There are marking criteria that we're given for marking the questions, and those criteria explain that you are expected to write an analytical and comparative answer to the exam question. So it's not just about sort of repeating the information that I've given you in this module. It's about taking what we've studied, reading around it, and offering your own perspective. So if you find a paper that relates to saccades, the kind of computational problem that the colliculus is carrying out, then incorporate that into your answer for the exam. You probably do want to plan which modules you're going to answer questions on. I think you have a choice, right? So understand the papers we studied in detail, but cast your net wider than that. Get a feel for the problems with the vector averaging theory and the vector summation theories for the colliculus. Dip into some of the papers cited by the Groh and the Goossens and Van Opstal paper, and do as much independent reading as you can find time for around the question of what the superior colliculus is for, what computational problems it solves, and kind of the surrounding tissue and how it's interacting with the rest of the brain. So there you are. Write an analytical comparative answer to the exam question that draws from the sources, and that is how you can get a really good grade in the exam question. Okay, I think we're almost there. Yes, that's it. So thanks very much, and I hope you've enjoyed the material.